I start recording right now. Okay, we're recording. So as I was saying, I'm Jaime Amaro Blanco. I'm the responsible for communication in the Institute of Marine Research here in Vigo, in Galicia. I'm going to just introduce a bit of the structure for the day, and then I'll just pass it on to Eva and to Chris to start with the conference. So the first thing we're going to do, as I said, is going to be, we're going to have a brief introduction of Chris Bowler and a brief introduction to the symposium and the Spanish Research Council by Eva Balsa, which is our Deputy Director for Research in the Institute of Marine Research. And then we'll have, for about 45 minutes, we'll have Chris Bowler, which we are delighted to have here with us. And his talk about Tara Oceans. As I said, that will last for about 45 minutes, and then we'll have our last 15 minutes will be a brief Q&A session. As you can see right now, no one's able to share their cameras or to share their audio. So please feel free to use the public chat that you'll see on the left side of your screens to ask any questions during the session and to say any comment that you might want to say. Afterwards, in the Q&A session, the last 15 minutes, we'll try and allow you to share your audio so you can ask yourself your own questions to Chris. If we see that gets messy or something like that, we'll block it again and we'll be the ones asking the questions. But if this is just a matter of the capacity of the platform, but we hope we have no problems with that. Furthermore, before letting Eva introduce Chris, we would like to ask you 3 brief questions to the people that are here today with us. This is just 3 questions that will allow us to know which audience we're reaching and who's here with us today. So you'll see a brief survey appearing on your screens. You just have to click the right answer. So the first one of them is this one here. So are you part of the CSIC, the Spanish Research Council? And now you should be seeing, okay, wow, that was fast. There you go. Great. I'll give you 5 more seconds. I mean, it's an easy question, right? So, okay. So we have 86% yes and 14% no. We have 2 people here that are not part of the Spanish Research Council, which is great. Okay, next question will be, are you working in Spain? And we have 3 options here. The first one is yes, specifically in Galicia, yes, but in another part of Spain, or no, I work abroad, I don't work in Spain. So I'll allow you to answer now. Here you go. All right. Okay. 5 more seconds. Okay. We have no one working abroad, so I'm guessing Chris is not answering the surveys, but there you go, right? And then the final question is, do you work in marine research? And we have also 3 options here. So one is, yes, I work in marine research. No, but I work, but it's just one but I do work in STEM. Or no, I don't work in science at all. So again, there you go. All right. A lot of marine researchers, as we expected, but you never know. Okay, there you go. So once this is finished, I'll leave the floor to our Deputy Director for Research in the Institute of Marine Research of the Spanish Research Council, which is Eva Balsa, which will be introducing the session. So thanks so much. Eva, the floor is yours.
Thanks, Jaime. Good morning, or I guess good morning because of the results of the quiz. And welcome to this new edition of Symposium. For those who are joining us for the first time, Symposium is an initiative organized by the research or the Marine Research Centers in the CSIC, the Spanish Council for Scientific Research, on the occasion of the Decade of Ocean Science for Sustainable Development as promoted for the United Nations. The idea of the symposium is to encounter, together with world-renowned researcher, as we are doing today with Chris, to discuss new frontiers in ocean science. So as Jaime mentioned before, please post your questions, write your comments in the chat, and we will address them at the end of the conference in the Q&A session, because the objective is, of course, to generate some discussion and debate. As Jaime said, my name is Eva Balsacanto. I'm the Deputy Director for Research of the Institute for Marine Research in Vigo, northwest coast of Spain, in Galicia, the northwest coast of Spain, just above Portugal. And we perform, and we are hosting this symposium today. And our research focuses mainly in three grand lines: the first, ocean and coastal areas. The second would be marine life and ecosystems, and the third would be biological systems and bioprocesses. We are also highly committed to social engagement, so we have our special units for that, and you can follow our activities in the webpage or through our social media profiles. We can write them down in the chat. As a researcher, I'm doing modeling in systems biology, so I'm really thrilled to introduce our speaker today, Chris Bowler, and his talk on Tara Ocean's ecosystems biology at a planetary scale. Our speaker today does not really require much of an introduction, because he's highly reputed and well known for all these his achievements. But I will try to, well, I will use a couple of minutes to read. I will read them because it's to read his or a summary of his numerous achievements. So Chris Bowler is Director of Research at CNRS Ecology and Evolutionary Biology section and head of the Laboratory of Plant and Algal Genomics at the Institut de Biologie de l'École Normale Supérieure in Paris. He graduated from the University of Gatwick, UK, with a degree in microbiology and microbial technology, and completed his PhD at University of Ghent in Belgium with a thesis related to stress tolerance in plants. He then pursued a postdoctoral study at the Rockefeller University in New York, working on phytochrome signal transduction in plants. In 1994, he established his own research group working on signaling in higher plants and marine diatoms at the Stazione Geologica in Naples, Italy, and in 2002, he took up his current position in Paris. He's ENVO member since 1995, recipient of the CNRS Silver Medal in 2010, and 2 advanced ERC grants in 2012 and 18, respectively. He received the Louis de Fondation Grand Prix Scientifique from the Institut de France in 2015, and he was also selected into the Académie d'Agriculture de France in 2018. And currently, he holds the chair in biodiversity and ecosystems at the Collège de France. He was also a fellow of the Radcliffe Institute for Advanced Studies at Harvard University in the period 2016 and 17. His major research interest is in understanding the response of plants and marine diatoms to environmental signals. In marine diatoms, he established molecular tools to assess gene function, and he has played a major role in coordinating the whole genome sequencing of several species. Using functional genomics, he has revealed the cellular response of diatoms to nutrients such as iron and nitrogen, or to different wavelengths of light, and to allelopathic infochemicals. Since the beginning of this year, he is the scientific director of the well-known Tara Oceans project, and as you know, this project has been performing a worldwide analysis of plankton community diversity and function in the global ocean using high-throughput DNA sequencing and cellular imaging approaches, and has already resulted in more than 200 scientific publications. According to the Web of Science, Bowler is currently among the 2% of most highly cited biologists worldwide, with dozens of thousands of citations to his works. We are really honored and thankful that you, Chris, could join us today in this virtual auditorium. And of course, without further delaying, please, whenever you want, the floor is yours.
Well, thank you, Eva Maria, for that nice, kind introduction. Way too exaggerated. Thank you anyway. So I'd like to thank you for the invitation to talk at your symposium, which is a great initiative within the UN decade. And I'm very sorry I cannot be with you today in beautiful Galicia. I hope sometime in the future we will be able to get together and enjoy some time together. So I think with that introduction, I will try and share my screen. Okay. Okay, I hope that is all okay. You can see that okay. Maybe I can even turn on my laser pointer. Yep, okay. So I'm going to give you an introduction today to the Tara Oceans project and some of the... Yes.
We can see the whole thing. I don't know if that's like the whole PowerPoint application program.
Okay, so let's see. I think I have to stop sharing and start again.
I go to, yeah.
Yeah. Okay, how about that?
Now it's better. Yeah, there you go.
Okay, good. And do you see my laser pointer? On the left-hand side?
Yeah, we can see it. Small, but it's there.
Okay, all right, great. Okay, so I'm going to give you a brief introduction to the Tara Oceans program. I guess most of you are aware of it now, and run through a few of our I think key results, novel results that are emerging from the project, and hopefully it will be a general enough presentation that at least some of the aspects will be of interest to your own work, to everybody who's listening. So Tara Oceans then began now more than 10 years ago as a 4-year expedition on the research schooner known as Tara, which is owned by the Tara Ocean Foundation in France, which is owned by Agnès B, which is a French fashion designer, who gives it to scientists on loan to do environmental research, basically. So we were able to, fortunately, use this vessel over a 4-year project from 2009 to 2014, aimed at exploring marine plankton ecosystems and trying to learn something about their sensitivity to climate change, induced modifications in the ocean. The project involved a round-the-world expedition, in fact 2 round-the-world expeditions, one through the global ocean and a second going around the Arctic Circle in 2013, and was run by Eric Karsenti, who was the scientific director during the last 10 years, in fact. And although it is a very much a French project, it is also very international, involving several partners around the world, including people from the CSIC, notably in Barcelona, our colleagues in Barcelona, Pep Gasol, Ramon Massana, Silvia Asinas, notably, was coordinating the CSIC activities within Tara Ocean. So I don't know if anybody from there is listening, but if so, hola Barcelona. So in addition to the science, we also had a lot of activities around the popularization of science through generation of media material, also generation of material for educational purposes, notably kids in schools, and also material to influence policymakers and try to give an appreciation of the importance of ocean ecosystems for the well-being of the planet by talking with policymakers and trying to get them to influence policy towards a better governance of the ocean. So I'm not going to talk about any of these activities today, but if you're interested to learn more, I'll be happy to talk with you during the questions or offline later on. So the science of Tara oceans then was all focused on exploration of the planktonic world of the ocean, the microscopic organisms that drift with the currents. And even though they are small, they're microscopic, that by no means means that they are not important. They're in fact incredibly important, as you will see in a second. On one hand, in terms of abundance, in terms of biomass content in the ocean, these microscopic organisms represent at least 2/3 of the biomass that is present in the global ocean. So that already gives an indication of their importance. And in terms of their functions, well, on the one hand, they are the base of oceanic food webs. So it's simply, if there is no plankton, there are no high organisms, there are no fish, there are no whales, there are no dolphins, etc. In the ocean, obviously. And then in terms of photosynthesis, these organisms are extremely important. They contribute as much photosynthesis on our planet as the plants and trees on land, as I will show, as we will discuss a little bit later. And so through this, they can take carbon dioxide out of the atmosphere. They generate biomass, which feeds the food chain. And by bringing carbon into the ocean, they also regulate climate by regulating, helping to regulate the amount of carbon dioxide in the atmosphere. Okay. Through photosynthesis, they also generate oxygen, which obviously also is important for the majority of organisms living on our planet. And through photosynthesis and through other processes, we know that plankton can both affect climate and are affected by climate change as well, which is a few things that I will discuss at the end of my presentation today. So we wanted to learn more about this mysterious world of microscopic organisms. Let's just quickly go through what kinds of organisms we find in this microscopic world. On the one hand, we have organisms called protists, or single-celled eukaryotes, or micro-eukaryotes, which have organisms that have all kinds of different shapes and sizes, as you can see in this little microscope slide here. We have organisms like diatoms in little boxes. We have organisms like dinoflagellates that look like little spaceships. And we have organisms that do photosynthesis. We have others that are parasites. We have others that are that have all kinds of other trophic life stories as well. So this is a very rich world of organisms already in the ocean. In addition to those, we have zooplankton, so the organisms that eat typically the protists. Many of these are multicellular, although they are still very small, like copepods, little shrimp type things that I'm sure you're all aware of. And then getting smaller, we have lots of bacteria and archaea that make up the prokaryotic world. And even smaller than that, we have lots and lots of viruses and giant viruses. So these can, for example, be present at up to 10 billion particles, viral particles per liter of seawater. And the prokaryotes can be present at up to like 1 billion cells per liter of seawater. So all these guys are pretty abundant once you get a microscope and start looking at them. Or through other tools, we can see that they're really quite abundant. They also have vastly differing sizes. You can see, hopefully, in this electron micrograph video, the range of sizes that are present in this microscopic world. So what we have here is a copepod, a zooplankton, with a diatom protist sitting on it, and a bacteria sitting on the diatom cell. Okay. So you can see the different sizes that we are going through, from the biggest copepod down to the bacteria here. And if we had a virus, it would be even smaller than my laser pointer, I guess. So all of this it equates to 5 orders of magnitude or something like that. So if you wanted to sample this range of sizes in an ecosystem, such as a forest ecosystem on land, it would be the equivalent of sampling everything from an ant all the way up to sort of a brontosaurus in size. So there's obviously no single, net that you could use to capture all of those organisms in a forest. And for the same reason, we're not able to capture all of these organisms in the ocean either. So we had to devise different methods to do that. And in the next, I'll get off from the PowerPoint and show you a video, or in fact, Jaime will show you a video that maybe shows you in more graphic detail how we did sampling on board. Let's see if that's going to work. Okay, yeah, I see that you can see it. I couldn't see it. So here you see some of the sampling that we've been doing on board, and how we, the scientific activity on board was really aimed at filtering large volumes of seawater and getting the microscopic organisms concentrated on membranes that were then put into liquid nitrogen and sent back to the labs. In addition, we had a few microscopes, a few cytometers on board to get some basic information about what kinds of organisms we found in the water column. And using the different sampling equipment we had, including a CTD rosette, we could sample down to like 1,000 meters in depth or so. Okay, so we typically had 5 scientists on board who would rotate every month or something like that. So in the end of this project, we had, 250 different scientists on board, mainly postdocs and PhD students, because it's pretty hard work. And it all worked out very well. So I think I need to get back to sharing my screen again, right? Yes, okay. Let me see. So just...
Chris, Marco in the chat said that he couldn't hear you properly. So if you can just summarize just a bit of what you said in the video, that would be great.
Okay. So I don't think you see my full screen now, right? So I will do it again. Okay. So you're able to see that?
Yep.
Great. Okay. I think I lost my pointer, but it doesn't matter. I can get it back. So yeah, just to summarize then quickly, if you didn't hear me well enough, yeah, we had 5 scientists on board typically at any one time, and they would spend their time doing the scientific sampling, trying to sample the entire water column from the surface down to 1,000 meters. In depth, using the CTD rosette to collect samples from depth, bringing it on board, doing a lot of filtration. That was the main activity of the scientists on board, filtering the seawater through different mesh sizes. I'm sure many of you are familiar with these kinds of protocols, getting the organisms onto membranes through filtration ramps and so on, and getting the samples into liquid nitrogen, ready for shipping back to the labs on land. So at the end of this exercise of 3.5 years or so, we had sampled at about 210 sampling sites around the world. You can see that on the map. And we had brought back about 40,000 samples for biological analysis, as well as a lot of other data sets and so on. So a lot of samples then to try and get through. And just in the same way as all of our activity on board was very much standardized, we always did the same thing no matter where we were. Also, the analytics back in the lab were also very much standardized, always doing the same thing. So the different size fractions that we collected on board, representing samples enriched more in viruses, or more enriched in bacteria, or more enriched in protists, more enriched in zooplankton. We would then take these samples and do metagenomics analysis. I will not go into detail what kinds of analysis we did, but this is basically high throughput DNA sequencing to characterize what organisms are there. And in parallel, we also had a lot of approaches aimed at high throughput imaging to see what the organisms actually looked like, because DNA information is basically one-dimensional, G's, A's, T's, C's. It's very informative, but it doesn't necessarily tell you what the organism looks like. And we wanted to also learn more about the morphologies of our organisms. So we spent also some time setting up high throughput platforms to image the actual cells, as I will show you in a couple of minutes. So all of this analytical pipeline back in the labs, performed by different people, notably the Genoscope for DNA sequencing and the EMBL in Germany for the imaging. Basically, all of this was aimed at trying to understand who is there, what do they do, with whom do they do it, and why is it important. It's a very simple question, but requiring quite a lot of analytics to try to answer that question. Over the years, we've done quite a lot of work with these samples. We've now published a lot of papers, sometimes in high-profile journals with the covers. We're happy to show that. And this adventure of publishing our work really began in now more than 6 years ago in the journal Science, which many of you probably saw. Where already we published the first foundational results from the project in 5 papers published back to back. We were very happy with this, that they gave us the front cover, they gave us the editorial, and that was a great achievement for us. So what we reported in those papers were the first primary analyses. From the prokaryotic world, we reported on the order of like 35,000 different prokaryotic taxa, the majority of which were already known, actually, containing around 40 million genes, the majority of which were unknown and still need a lot of analysis to understand what they do. In another paper, we reported the viral communities in the ocean, 5,500 different types of viruses, only 39 of which were previously known. And we reported the order of like 130,000 genetic types of protists, unicellular eukaryotes. This number represents more than 10 times the number of formally described species of marine eukaryotic plankton. And about 1/3 of these we cannot assign to any known taxonomic group. So again, showing our ignorance really in terms of understanding life in the ocean. How little we know about these organisms. So subsequent to that first splash of papers from the project, we've reported additional resources from the project. A couple of years ago, an atlas of eukaryotic genes now, containing 116 million genes, mainly from protists. So this adds to the 40 million genes we have from prokaryotes. So we have like more than 150 million marine genes now available for people to study. We also generated single cell genomes from diverse, abundant, but uncultured protists. So this is extremely important to get information about what these this unknown component of the ecosystem does. These are organisms that we don't have in culture, so we really know nothing about them. But by sequencing their genomes, we can start to understand what roles, metabolisms these organisms actually have. And then finally, another resource that we published more recently is concerns the viral communities. So now we've have up to 200,000 different kinds of viruses described from the ocean, including from the Arctic Ocean. So this goes up from 5,500 that we reported in the science papers. So these are the resources that we generated from the project. Now, what can we do with them? And what can we do with this data? Let's just go through some very simple analyses of what we can look at and where we actually got surprises even from these very simple studies. So a first question that you could ask is, which organisms are the most abundant organisms in the plankton communities? We weren't sure what we expected to find, but we were certainly surprised to see that radiolarians are unexpectedly abundant in the ocean. In particular, two groups of radiolarians known as Colodaria and Phaeodaria. This we really found mainly through in situ imaging, not through genomics, I have to say, through in situ imaging. To reveal the abundance of these oftentimes delicate, fragile organisms. So we've known about these organisms for more than a century. These are some amazing images from drawings from Ernst Haeckel, like 150 years ago, but we never knew their abundance, their importance in terms of abundance in the ocean. So that was one surprise that came through from the data sets. Another surprise were the organisms that were the most diverse group of organisms in the ocean. We were surprised to find that these are diplonemids. You can be forgiven if you've never heard of diplonemids. Most of us had never heard of diplonemids either, I have to say. But we were very surprised to find an incredible diversity of this group of eukaryotes in the ocean. We don't really know much about diplonemids. They are sitting within this group of eukaryotes called Disci-cristata in the eukaryotic tree of life. And they're close to trypanosomes, leishmania kinds of organisms. So we hypothesize that they may be parasites or something like that in the ocean. But because we have so little information about the lifestyles of these diplonemids, we really don't know more about them than that. So those were already some big surprises in terms of the most abundant organisms and the most diverse organisms. So we can dig deeper and imagine that, expect that we'll find even more surprises as well. So something else that we can do with all of this huge amount of data that we have is sort of put all of this data together and try to see which organisms interact with each other or which organisms never interact with each other. So just sort of by guilt by association in all of our different samples, we can look for organism-organism co-occurrences or organism-organism exclusions through our data set. So we can try to define the sort of social network of the ocean plankton, if you like. This was a very giant compute several years ago. Took a lot of compute power actually to generate this. But here is the sort of basic kinds of results that we get from this kind of global study, where each point here is a different organism. In blue, they're different eukaryotes. In pink, they're different bacteria, and so on. And then if we have green edges between them, that indicates that these organisms co-occur with each other very commonly in our samples. And organisms that are connected by red lines between them are organisms that we never find together in the same sample. So implying that there may be some sort of mutual exclusion or something like that. So there's a lot of information in here. I'm not going to go into any detail at all, but just the one point that I wanted to make here was by looking at interactions in terms of biotic interactions, so organism-organism interactions, and comparing that with interactions between one organism and the environment, an environmental parameter, we actually found far more taxon-taxon associations than we found taxon-environment associations. So implying that biotic interactions are perhaps more important than abiotic interactions in building this network of plankton. that defines their communities. So perhaps what this is telling us is that bottom down processes, biological interactions, are perhaps more important than top down interactions are perhaps more important than bottom up interactions that are governed by nutrient availability, temperature, and so on. So that was an interesting finding that came out of these studies at this very high top level of looking at interactions. So this is there's a lot of information in this interactome, but at the end of the day, this is just a computer-generated scheme, right? We don't know how much of this is actually means something in terms of interactions. Where that kind of question can be helped is through our imaging platform, where, as I mentioned, we set up these high throughput imaging platforms that allow us to see the organisms at different scales of resolution. Here are three organisms where you can see their sort of extracellular structures, their exoskeletons. You can see the intracellular structures as well. These are all, just a few, maybe 100 microns in size or less. In the top left, you have an acantharia. In the top right, you have a dinoflagellate, and in the bottom, we have a diatom. The blue is the nucleus. You can see the elongated nucleus in the diatom. The red are chloroplasts, and so on. So by combining our interactome with this kind of imaging data, we can spot interesting interactions. And here are two interactions just to as visualization tools really for my presentation, where you can see specific interactions revealed through the high throughput microscopy approach, which is subsequently verified in our interactome analysis. So here we have a chain of diatom cells being infected by Vorticella, a ciliate, quite clearly sticking to the outside of the diatom chain here. And then on the right here we have a chain of diatom cells with each one containing nitrogen fixing bacteria that are labeled in particular in green inside these cells. So this is where we can really begin to think about using these single cell approaches to address biogeochemical questions. In this case, nitrogen metabolism, nitrogen fixation in the ocean, which has enormous, obviously, biogeochemical implications. Our knowledge of nitrogen fixation in the ocean is a bit fragmentary because we have different studies from different places, not always aimed at looking at the same organisms and so on. But with Tara Oceans, because we have this global data set and we have the imaging and we have the genomics data, we can look at the relative abundances, the relative contributions of different kinds of nitrogen fixing symbiotic associations in the ocean and compare, get a feel for their relative importance. So here is a study where we've done this in collaboration with Rachel Foster in Stockholm. And here we see single cell, free living, non-chloroplast diazotrophs, non-photosynthetic diazotrophs. We see their abundance in the Pacific Ocean. Here we see trichodesmium filaments, particularly strong in the southern Indian Ocean, and so on. So this allows us to get a global picture of the relative importance of these different kinds of nitrogen fixing organisms, which we can then use to teach us more about nitrogen fixation in a biogeochemical setting in the ocean. So that's nitrogen. Let me say a couple of words about carbon. First of all, photosynthesis. I guess you all know that photosynthetic organisms in the ocean contribute 50% of photosynthesis on our planet. So these phytoplankton, even though they're microscopic, they are as important as the plants and the trees on land, amazingly so. These are the kinds of organisms that we know about that are doing photosynthesis in the ocean. We have prokaryotes on one side, Prochlorococcus, Synechococcus, nitrogen fixing cyanobacteria like Trichodesmium, and then a range of eukaryotes, including green algae, dinoflagellates, diatoms with their silica cell walls, coccolithophores with their calcium carbonate cell walls, and a range of symbiotic associations. So again, through the global data set of Tara oceans, we can look at all of these at the same time and look at their relative abundance and their relative diversities with respect to each other. To do this, we have to cross the prokaryote-eukaryote divide, which is a problem in ribosomal RNA-based approaches. But what we've done in my lab, an Argentinian postdoc, Juan Pierella, has set up the use of PSBO, which is a photosynthetic gene, a single copy gene present in all photosynthetic organisms, prokaryotes and eukaryotes. And so using this PSBO gene, quantifying it in different fractions, we can see sort of the relative distributions of different kinds of phytoplankton, and also their diversity in terms of Shannon index shown in the color scale. So we see, you know, diatoms, for example, they like it cold. They like the Arctic region, Southern Ocean region. Other kinds of organisms like the picocyanobacteria, they prefer the lower latitudes, as we know. And we can see also through here the typical latitudinal gradients of diversity, with diversity being strongest in the equatorial regions and getting weaker as we go to the poles, in this example of haptophytes, for example. So it's nice to be able to quantify all of these different organisms on one page, if you like, on one slide. And that gives us a feel for the relative importance of each of these. What we also appreciated from the studies that we have done is the importance of symbiotic interactions in photosynthesis. So these are photosymbionts, typically involving calcified forams or silicified radiolarians, forming symbiotic associations with algae, different kinds of algae, which we see also to be particularly important in the ocean, perhaps underappreciated up until recent times. So this is how we can look at these different organisms, look at their relative contributions, relative diversities, and so on. What we can further do is look at their relative contributions to photosynthesis, photosynthetic activity, to net primary production in the ocean. So we can do that essentially by sort of comparing abundance of organisms, organismal groups, with respect to different environmental parameters. So this is basically what we're showing here on the X and the Y axes. And in terms of photosynthesis, the measures that we have on the one side, we have net primary productivity, which is calculated from remote sensing observations from space. And then we also have chlorophyll measurements from HPLC data, samples that we collected, or chlorophyll fluorescence from the water column as well. So we have these parameters that are particularly important for photosynthesis, and we're interested here to see, well, which organism correlates the most strongly with these photosynthetic parameters. And what we see is it's this guy here that is the most strongly correlated, which turns out to be the diatom. So on the global scale, this simply shows that the diatoms appear to be the most important contributors to primary productivity in the ocean. Not necessarily a surprise, but obviously from here you can dig down and see where are they important, in which regions, and so on. So photosynthesis is one side of the biological carbon pump in the ocean. The other side is the export of carbon, export of particulate material down to the ocean interior. So this is an extremely important process in climate regulation. It involves uptake of 40 to 50 billion tons of organic carbon per year. This is, I think, about twice the amount that plants will use, carbon dioxide will use on land each year. And of this organic carbon that is generated, a small amount will sink into the deep oceans, and an even smaller amount will actually make it down to the sediments, and over millions of years will generate the oil and the gas fossil fuels that we are so eagerly burning today. So we have some basic information about how this biological carbon pump works in terms of decomposition of material, the importance of microbial activity, the importance of grazing, relationship between respiration and photosynthesis, and so on, through many decades of research focusing on the biological carbon pump. What we tried to do to contribute to that was to see, which plankton taxa are the most strongly associated with this biological carbon pump. So we could do that because on the one hand we had data on particulate content in the water column at each Tara Ocean station. So we imaged particulate material in the water column from 0 to 500 meters in the water column, giving us these global maps where you see some areas are pretty poor in carbon particulate material in the water column, whereas other regions are very rich, like the Southern Ocean, for example. So we can take this data and we can compare it with our interactome data that I introduced you to before, to sort of see, well, which organisms appear to correlate the most strongly with higher carbon export, higher particulate material in the water column. So we can go from these rather simple representations of plankton and plankton activities in the water column to a more sort of graph-based visualization, where we can use these methods to identify specific kinds of plankton that are sort of central to the functioning of this whole network of organisms that appears to correlate particularly strongly with enhanced carbon export. And this led us to some organisms that we expected to find, like radiolarians and dinoflagellates, as being particularly important, but also some small guys like the Synechococcus, Picocyanobacteria, and even smaller viruses, the Synechophages, which infect Synechococcus. These appear to be particularly important in that they're associated the most strongly with increased carbon export activities. So these are the sorts of things that we can do using this data set to explore particular biogeochemical processes, in addition to analyses of biodiversity and so on. Now, in the last 5 minutes or so of time that I have, I'd just like to address one very important topic, obviously, which is climate change, and what Tara Oceans is contributing to try to learn more about climate change associated consequences for ocean ecosystems. So, you have all seen this this data of how our planet is changing over the last several decades. Human-induced warming, planetary warming, has reached approximately 1 degree C now, above pre-industrial levels. Carbon dioxide concentrations in the atmosphere have been increasing steadily. They're now close to 420 parts per million, sadly. Atmospheric concentrations of carbon dioxide are higher today than they have been for any time in the last 800,000 years. We have very solid evidence from analysis of Antarctic ice cores. Sea surface temperatures have increased by almost 1 degree now since 1900. The data now begins to be very clear in showing these trends, these temperature anomalies. And sea levels have increased now by 19 centimeters or so in the last 100 years. And all of these trends are obviously expected to continue into the future. The summer Arctic sea ice has lost about half of its surface in the last 50 years or so. And the seawater is steadily acidifying. Surface pH has decreased so far by around 0.1. And again, the data is very clear now showing these trends. So can Tara Oceans contribute to learning more about what the impacts of these changes will be on marine ecosystems? I'm going to show you three examples. One relating to evidence for the effects of ocean acidification. The second, projected changes in plankton biodiversity by the end of the 21st century. And the third, the uniqueness of Arctic plankton communities in particular. So first, in terms of ocean acidification, there's a huge literature on the impacts of ocean acidification these days. One of the issues that we have in looking at ocean acidification is it's difficult to find long-term trends because we don't have data from the ocean, 200 years ago, 500 years ago. But one interesting data set that we have is from the HMS Challenger expedition, which is considered the first oceanographic expedition, which was launched worldwide expedition in the 1860s. And they collected a lot of samples, many of which were used by Ernst Haeckel for his beautiful drawings. And so what a group of researchers have done in the Natural History Museum in London is to compare some of the Challenger samples with some of the samples collected by Tara, almost 150 years later. And they were interested in particular to look at foraminifera calcification, these little organisms that have calcified shells that we know to be particularly sensitive to pH. The generation of these shells is sensitive to pH. And what these researchers did was to compare the thickness of the calcified shells of these forams collected by HMS Challenger with the thickness of these shells in samples collected by Tara in the same location in the Pacific Ocean. And you can see very clearly that the thickness of these calcified cell walls has really diminished very impressively over the last 150 years or so, perhaps implying here one impact of ocean acidification on life in the ocean. So there is one example that has emerged over recent years, which I think is a nice example to show. Something else that we've tried to do is to use the data that we have from Tara Oceans from today's ocean, right? Ocean at the beginning of the 21st century, to try to project how plankton communities may change in the future. So we initially did this by generating global maps of diversity of different kinds of plankton in our samples. Here we have 6 examples: copepods, photosynthetic eukaryotes, and so on, showing typically the latitudinal gradient of diversity, with highest diversity in the tropical regions and dipping off towards the poles. And with this data, we can take this data and put it into the projected future ocean as predicted from the IPCC climate models, in particular CMIP5 model compilation of models that were put together by the IPCC, where they have projected sea surface temperature changes by 2100, changes in primary production, and so on. So we can take this data and see how our organisms are going to behave in this future ocean in terms of diversity. And here we see the projected diversity anomalies that were generated from this analysis for each of these 6 different plankton groups. So what we're plotting here is anomalies. So anomalies meaning increases in diversity in red and decreases in diversity in blue. You can see this for the different groups, and you can see they're quite complex patterns globally. But the interesting thing to see, I think, is in terms of latitudinally, where we compare these projected anomalies with very important ocean processes. For example, carbon export, the activity of the carbon pump in the ocean, our average fish landings shown latitudinally, and the current sort of activity we have of marine protected areas that we have globally. You can perhaps see that better on a map here, where we have these diversity anomalies for the different groups, and the relation of these anomalies that are projected on these maps of carbon export, of fish catch, and density of marine protected areas. And so you can see that the impacts of these projected anomalies are likely to impact these very important biogeochemical, economic, conservation activities in the ocean, which is something that we need to think about as we move into the future. And then finally, let's just look at the Arctic biome. I haven't talked much about the Arctic, but the Arctic is, of course, extremely important environment. Is changing faster than anywhere else on the planet. Tara sampled at several sites around the Arctic in 2013. And from that data, we could add that data to our global interactome data set and sort of generate a new interactome of the organisms to see how organisms interact with each other computationally in silico when we combine the Arctic data set with all of the other data sets. And what we can see quite clearly from this analysis is from the color coding here that the organisms isolated from the Arctic really separate out as a different organization from the global interactome generated from the rest of the sample. So this really shows that the Arctic interactome really behaves as a distinct ecosystem compared with the rest of the ocean. Okay, so that was a very clear picture that emerged from this analysis. What we then attempted to do, colleagues in Nantes, Sam Chaffron and Damien Eveillard in particular, who led this work, was to challenge this interactome to explore its sensitivities to changing the environmental parameters. So they attacked the interactome by changing temperature, by changing nutrient content, by changing pH, and they saw how the interactome disintegrated basically by attacking the network, by increasing temperature or other manipulations. And what they found from this analysis is that the polar community is particularly sensitive to changes in temperature. So really implying its sensitivity to changes in temperature, very specifically in this Arctic context. And then finally, we can take this Arctic specific network of interactions, and we can try to define organisms which we could consider perhaps Arctic indicator species, or perhaps keystone species through exploration of their keystone indexes. How central are they to the structuring of the whole network? And this, again, from this analysis, it emerged that the plankton within this Arctic network are particularly important in terms of their keystone index with respect to other taxa from the other from the larger interactome. So those are three examples there quickly of the kinds of things we can do to explore the sensitivity of ocean ecosystems to climate change, in this case using the Tara Oceans data set. And I think I will wrap up here and just leave you here with a few take-home messages of what I tried to show you here, and then just go straight to my acknowledgement slide. A lot of people are involved in this work worldwide, including the CSIC in Spain. We're very proud to have working with us, notably the ICM in Barcelona, many other labs as well. And thank you to our funders and our sponsors. And just let me mention that Tara is now at sea again, doing a microbiomes mission, looking at in even further detail at this microbial world in the ocean. So thank you to all of these participants in the project, and thank you to you for your attention. I will end here. Thank you.
Thanks so much, Chris. That was amazing. Really, really interesting talk. And I think we'll all agree on that. And yeah, thanks like on behalf of the whole of CSIC, especially ICM and all the marine research centers, and also our research center, which is the Institute of Marine Research. And now I think we have some time for questions. So people have been a bit shy on the chat. They haven't asked a lot. But what I'm gonna do now is I'm gonna let them open their microphones. And once I do it, if you want to open your mic and ask the Chris directly, you'll have to first press on the phone button you can see there. I'm gonna show you right now. So wait a moment. So this is the next. There you go. So first you will have to press the telephone button, and then you'll probably have to allow the audio, and then you'll be able to mute or unmute. Okay, so I'm gonna allow you now to do that. Beware that you have to press that button.
In the meanwhile, may I ask one question?
Yeah, please, go for it.
Thanks, Chris, for the very nice presentation. And I was thinking in the case of, let's say, terrestrial microorganisms, and in the context of systems biology, or with the aim of understanding metabolism of those microorganisms, there are a number of let's say, models. So E. coli would be one model, and Saccharomyces cerevisiae would be another model. And there are communities around those organisms building what is called consensus metabolic models built from the genome sequences. If you had to choose a model for those microorganisms we have in the ocean, would you select one of those radiolarians, or would you select something else to build, let's say, a community around this kind of approach?
Yeah, it's a very very difficult question to answer, because one of the important aspects of understanding how the marine ecosystem works is taking into consideration the complexity of the whole community of organisms. So as soon as you want to think about metabolic modeling, then you begin to reduce the complexity of the system, which already takes you away from the natural way that it works. But of course, we have to try to do that. I think we have to try to break down the system in order to try and build it up again, as you are doing. I fully agree that is what we have to do. And as you say, E. coli, yeast are being used to do this in other contexts. In the ocean, there is some work with Prochlorococcus, with Synechococcus, which is moving forward quite well. I'm also aware of work being done with diatoms. In that case, they're using the model diatom called Phaeodactylum tricornutum. So Phaeodactylum is great in the lab because it grows really easily. You know, even students are not able to kill it. It's sort of resistant to students, undergrads, and
that is
which makes it
super
very
resistant.
You can do genetic transformation, so you can use CRISPR-Cas9 to knock out genes, to assess function, to look at metabolic consequences, and so on. So people are beginning to use Phaeodactylum to try to build these metabolic models, with the hope that that will tell them something about the diatoms in the ocean, which I think it probably will. It will help us to understand something about diatoms, but still we have to realize that Phaeodactylum is not a good representative of diatoms from an ecological context, because it's almost never found in the ocean. So I think in all of these metabolic modeling studies, you do need an organism that you can manipulate in the lab to test hypotheses. If you come to a specific metabolic pathway of being a key metabolism, phosphonate metabolism or something, you want to be able to demonstrate that by knocking out the pathway and showing that it has a major effect on the cell. And so I think we do need these simplifying approaches of using model organisms in order to help us address the whole global complexity. So there is a space for both, I think, and I think diatoms is one option. The problem with the radiolarians are that we don't have genetic transformation systems, we don't have good genomes yet, and we don't really have a model radiolarian organism that is undergraduate resistant.
Okay.
You know, these things are so sensitive in the lab, you know, they die after a few days, a few weeks, and so that's already a big challenge to get these organisms cultured in the lab and usable for genetic manipulation.
Okay, I understand. I don't know whether there are any other questions.
We have some questions. Apparently, we're having some problems unblocking people to allow them to talk. So
Okay,
I'll read some of the questions we have in the chat. The first one is from Ramon Masana, who congratulates you on your talk. He says it was an impressive amount of work and coordination. And he asks, what are the future sampling plans with Tara?
Yes, so as I said, Tara is now in the middle of this microbiomes mission. She is currently in the Caribbean, being refitted to begin a major new sampling campaign down the South American coast over the next months. And so the plan is to study in quite some detail the river Amazon plume system in terms of consequences of biodiversity and so on. Then there will be some studies around the Malvinas area of Argentina to look at the coccolithophore blooms in particular later towards October, November kind of time. Then there will be some work in Antarctica, looking at gradients of nutrients coming off of icebergs, trying to understand those. And then Tara will go over to Africa. We'll go up the West African coast, looking at the upwelling systems in Mauritania, in Benguela, to come back to Europe in 2022. And then there will be a Europe-wide sampling expedition run together with the EMBL in Germany, where Tara, the project will be basically looking at the land-sea interface, consequences on biodiversity and human impacts in ocean ecosystems and so on. So that will be happening between 2022 and 2023. So a lot of things being planned in the coming years. And I know that that Sylvia is also involved in some of these activities as well, and organizing events in Barcelona and so on for sampling. So together with the EMBL outstation in Barcelona, of course. So still lots of exciting things coming forward in the future. The idea is to enhance imaging approaches, to enhance sequencing, long read technologies, for example, more single cell work, etc. So yeah,
you walk the board, I guess. You have plenty of things to get distracted with.
yeah.
Okay, we have another question then from Josep Maria. I don't know if you can activate your
I can say.
There you go.
Hi,
Great.
Chris. How are you? That was a great overview as usual. I mean, that's.
Well, thank you for your contribution, and Ramon's.
And I'm wondering. I mean, there is indeed clearly a global large success story about Tara. So I wonder if you can tell anything that can be passed to other scientists, and especially to managers and political advisors and so on on how to run something as big as this and with so many people from so many countries, and it ends up being successful? I mean, any lessons to be shared?
I mean, yeah, I mean, I could talk for hours about that. Yes, I think it was very much a bottom-up project. Normally, when we do science, right, we first have an idea, and then we try to get funding for it, and then maybe 5 years later we're successful in getting money, but then we've lost interest in the project and we've moved on to other things. In this case, we really didn't have any money. We just had a dream. We just had the real passion to want to do this project, to bring different people together, and try and make it happen, make it successful. So different people with their motivation for the project were able to secure enough funding to get the project started in the first place. But when we left, when we started the project, we only had funding for like 1 year of ship time. So we didn't know where the money would come from to keep the boat moving for the following 2 years, which was obviously a big risk, but we decided to do it anyway. And we made a few calculations in terms of how much money we would need for DNA sequencing of all the samples that we were collecting, and we estimated it would be several billion euros. Totally impossible. But while the expedition was going on, the price of DNA sequencing went down almost 1,000-fold, actually, as new technologies came along. So that was very much a game changer. So it's certainly a combination of factors that helped. It was the motivation of the individual researchers, including you guys in Barcelona, of course, the team that we had, that were really motivated to make the project a success. The leadership of Eric Karsenti was absolutely essential, of course, and logistically, having German people doing the logistics was really crucial. Absolutely.
Great, many thanks.
Yes, so that helps a little bit with a few considerations.
Thanks a lot. I'm sure it does. Now we have another question from Jose Pintado, which I think is going to be able to turn on his mic as well. So, Pepe, you're muted now, but you have to unmute yourself and you'll be able to talk.
Okay, I had to get out and get in again to get my microphone. Well, thank you for this brilliant conference, really interesting. I wanted to ask the question because I work with olobionts, and I want to know if any, marine microscopic olobionts, as fish or mollusks, can act as dissemination vectors of microbial taxa. So have they been considered in any global study?
That's a really good question. My short answer, I guess, would be no, they haven't really been considered. But I think we have to consider them more seriously as vectors of transport, because these organisms, they have their own microbiomes. Copepods, fish, whales, they all have their own microbiomes. They transport things around. And again, technology is improving, and through eDNA technologies that are slowly becoming more standardized and more, I think, able to give us quantitative data, we have more possibilities to populations of different macroorganisms at the same time as tracking these microorganisms. So through exploration of eDNA, looking for traces of whale, dolphin, fish, whatever DNA in the samples, that can help us do that. So I'm sure this is something which will be looked at more closely in the future. Certainly, I know colleagues in ASTI in Bilbao are very interested in this aspect. Nayara Rodriguez Espeleto, for example, is working quite hard on that. So I think that's something. It's a very important point. It's very important that we try to relate this microscopic world with the larger organisms, and yeah, hopefully we will start to see that happening in the next years.
Thank you.
Okay, and we have one last question so far, which is from Celia, which I don't know if she'll be able to open her mic, but probably not. So I'll be asking the question. Celia says, after Tara, did you detect a need of experts in taxonomy of some groups?
Oh, yes.
Oh, yeah.
Yeah, I mean, this is a real problem. Yeah, but another thing that we have seen during the lifetime of Tara is that more and more taxonomists have retired. They haven't been replaced by others. And so, yes, I mean, we are able to these days generate, huge amounts of genomics data, but the interpretation is really the bottleneck. The interpretation of gene function on one side and the interpretation taxonomically on the other. And we have so much to learn still about the evolutionary relationships between these different micro eukaryotes, their different activities, their different life histories, and we have fewer and fewer people to help us with the really basic taxonomical assessments. So this is a problem that doesn't seem to be resolvable in the coming years, sadly, and it's going to be a problem for us as we collectively lose that knowledge, taxonomic knowledge, and that expertise. So we have to try and really push the appreciation of funding agencies that taxonomy is a key scientific activity, and that it is just as relevant today as it was 200 years ago, and is even more relevant than it was 20 years ago. So yeah, that's an important message to pass, absolutely, and that is a bottleneck right now.
Yeah, I mean, Celia is also asking now, like, how could we improve the situation about taxonomists? But I guess you kind of answered that now. I don't know if you want to add anything to that.
Yeah, I mean, I think if we all pass the message, if we collectively pass the message, we have more chance that that we will get funding for a new generation of taxonomists that can, appreciate the morphological aspects, and that can go beyond G's, A's, T's, and C's in DNA sequences.
Great. Okay, so we were into the end of the Q&A. I would like to ask one last question that we have on chat, which is from Magda. I don't know if Magda is going to be able to open her mic. I think she won't. So I'll also be asking this question. So Magda says that congratulations for this interesting work. And she asks that in the networks of networks you showed, it seems to be some key node species. Which kind of species are these node species? Mainly photosynthetic species?
Yeah, that's a very interesting question. And I would say in general, no, they are not necessarily photosynthetic. They are different kinds of organisms, often organisms that we know very little about. Marine alveolates, for example, marine ochrophytes, that are not very well studied. These often appear to have, central positions within these networks. So again, it indicates that we really have to learn more about the roles of these organisms in the environment, if they are symbiotic, if they are parasitic, if they are able to, you know, change their lifestyles from interacting to being pathogenic. That really, again, opens up the need to have some basic knowledge about the life histories of these organisms, which we currently don't have. So, yeah, it's way the ocean is way more complex than just diatoms and copepods and prochlorococcus, if you like, which, you know, the organisms that have the main, you know, they get the biggest news in the press. The ocean is much more than diatoms and copepods and prochlorococcus. Maybe that's an important message to leave with. Yeah. There's lots of organisms that appear to be really important that we know very little about so far. So
we have a lot to still to learn and to discover about the ocean, and that's a really great message to take home, I think. And that we know that we're working for something that is great, important, and we have a lot of work to do, so we can keep on doing that.
Yeah.
Okay, I think with that, if we don't have any further comments or questions, I think we can close the session. I would like to thank you again, because it's been great to have you both for the conference and also for the way you answer the questions. I think it's a great way of answering. So thank you so much for being so clear. Before you all leave, I would like to remind you that there's going to be another symposium next month. It's going to be the 7th of July. And this time, the center that is going to be organizing it is the Institute of Marine Sciences of Andalusia, which is also obviously part of CSIC. And we have Dr. Diego Macias with a talk on marine sciences supporting policy making, the example from the Joint Research Center of the EU Commission. So don't miss that. It's going to be on YouTube. It's going to be really interesting. So yeah, pay attention to social media, ICMA social media overall, to know when it's going to be and where, and the link and all this stuff. With that said, thank you so much to all of you for being here. Thank you so much, Chris, again. Thanks, Eva, for being here as well. And I think we can close the session now. So thanks a lot.
Thank you very much. Keep safe. Bye-bye. Bye-bye.
Thank you.
Bye-bye.
Bye, Chris. Bye, everyone.